Good morning, everyone. Welcome to Thursday morning medicine grand rounds. Got a couple more people just joining us, but it's 8:02, so I think we're going to get started. I want to make sure that everybody knows, please mute your microphones so that there's no feedback during the talk, and then we'll have time at the end for questions when you can unmute if you have a question. Today we have, all right, let's wait till everybody gets in here. Some late people, but okay. So Anas Bizanti, who is one of our third-year medicine residents and an associate chief, is going to be speaking with us today. Anas did his medical education at the University of Tripoli, Faculty of Medicine in Libya. Before that, however, he trained in petroleum engineering and has a master's from the University of Central Florida in nanotechnology. So an engineer and a physician. And today, Anas is going to be speaking with us about improving immunotherapy through nanotechnology, a very important topic, especially in this era of vaccines, liposomes, and other things that have come on board to hopefully get us out of this pandemic. So we welcome Anas. Look forward to a very interesting talk, and I'll turn it over to him.
Thank you very much, Dr. Palmers, for this great introduction. And welcome, everyone. Good morning, and thank you for joining us at this time. We'll get down to it. We'll be talking today about immunotherapy and how nanotechnology did an impact in improving immunotherapy and cancer therapy in general. So as Dr. Palmer said, I had the pleasure to do 2 years of master on nanotechnology at the University of Central Florida. It was an interesting time where I had interacted with multiple people in different specialities, from chemistry and physics and science, all of them trying to advance medicine through the nanotechnology. I don't have any financial disclosure today or conflict, but I would say that my son had helped me with the slides. So during those 2 years in the University of Central Florida, we learned how to manipulate and how to see nanotechnology. And during that time, it was interesting to see what people who are not from a medical background, how their thoughts and how their training trying to advance medicine. And I saw a gap there between practicing and what is going on in the research. Definitely, they are one of the smartest people I have met. And then I come here into the medical practice, and I also saw that there is a gap as well, at least in this specialty, in terms of nanotechnology, and physicians are not really aware about the nanotechnology, which inclined me to do this talk. So I wanted to do the opposite, given this is not the unorthodox topic. I went ahead and asked my colleagues and my mentors here at St. Agnes, if you hear this lecture, Improving Immunotherapy Through Nanotechnology, just hearing the title, what questions do you have? And this is some of the questions. What's nanotechnology? What are the types of nanotechnology? Is nanotechnology already being applied, or just something in the clinical, in the research phase? And how bad is altering our immunity can be toxic? So that's what I would be trying, and that's how I made my agenda today. For the first part, we'll be talking about introducing nanotechnology and how those characteristics will change the practice. Then I will be talking about the current advance and the current medication that have been altered by nanotechnology. And finally, how does it alter our practice as physicians or specialists? Why do we need to know about this, and how would this really alter our practice? So we'll go ahead and start just how important this subject is. In 1990, there was 0 publication about nanotechnology. Last year, there was more than 20,000 publications, just telling you how much we're going, expanding. There was less than 10 million in funding in 1990. Now we're going into more than $2,000 billion in research and development from funding and grants last year. So definitely an expanding branch. So what's nanotechnology? The word nano itself is just, it's basically 10 to the power of -9 of a meter. So there's a million 9 zeros actually in one nano compared to the meter. Definitely you can go with smaller pico and femto, but we are not able to see at that level yet, but we are reaching an atomic level. Richard Feynman, this is the guy who actually first had the vision to nanotechnology. In 1959, he wrote a paper called "There is a Plenty of Room at the Bottom," one of the fantastic papers that I have read. In 1959 at Pasadena, this guy had written and had a vision of how nanotechnology could be done and what could be the application. Keeping in mind that at that time, when Dr. Feynman have written this article, we only had the light microscopy. In 1986, he actually had another paper and he won the Nobel Prize in Physics, a tiny machine about his theory in quantum mechanics. He have actually spoken in his article. He said, what if I could have written the whole encyclopedia on a pen in a pen head? And a pinhead is a 1 million nanometer wide. And then he further discussed how if you go to the side of sheet of a paper, this is a 75,000 nanometer. We're talking too large here. And today we are able to go into much, much smaller than that. And just to tell you how big is a nanometer, so 10 hydrogen ion atoms laid side by side is only 7 nanometer. We're able now to manipulate, create stuff down to the 2 and 3 nanometer. The double-strand DNA is a 2.5. Red blood cell is 7,000. So the red blood cell is huge to what we deal with. And when we had the light microscopy, keeping in mind the light wave is actually bigger than even a nanoparticle. So at that time, it was only an imagination. We couldn't even really see them until we started to have our eyes and our hands to manipulate them, if sort of say. So this is our hand in nanotechnology. This is what we call an atomic force microscopy. This is the first time we were able to touch and manipulate things on a nanoscale. It's a very simple device, but the application is interesting. So this is the cantilever, and it have the tip of the cantilever is actually 1 nanometer. And we start moving this on a sample size. This cantilever will actually go into the X or Y or Z according to the structure. And by having a laser here pointing out according to the movement, then it will be reflected, and we will be knowing the picture in terms of a nanoscale. This is our eyes, the scanning electron microscopy. This is how many cantilever that I have ruined during my training. So now we have the eyes and the hands to go into a nano level. We started to see a new world. This is the world of nanotechnology. We were able to go into an atomic level, and now we actually even recently are even able to manipulate and move atoms from one side to another. This is a huge thing that we have been able to do within the last 20 years, and definitely had a huge impact on us. So, but what's interesting about nanotechnology is that the smaller the size we go, we had the new properties, but by just going smaller. Actually, the whole mechanic laws have changed, although the fundamental do hold. But the physics and the chemistry did changes when we just went into a smaller size. Let me explain this very easily. If you could look at a 1 centimeter drop of water, it have a 1 centimeter cube actually have a 6 centimeter surface area. We have a surface of 6 centimeter. If we start dividing into smaller and smaller unit, if we go into the nano size for the same total volume, now if we go nano and we divide it into nano, we have a 60 million square meter square centimeter. We are increasing by 1 million times our surface area in just 1 centimeter cube. The application of that is it's tremendous, and actually we're already applying. This is stuff that we have in our hospital. This is in every floor in St. Agnes. We have a topical agent of silver nanocrystals, and they're using just that small amount of silver to cause with how much surface area, and it have this antibacterial activity. Another thing that was very interesting to see when we went smaller is how the fluorescence in the quantum dot, they change differently. The color and the fluorescence do change differently. Very simple here. All of those compounds are gold. And we're just changing the size from 1 nanometer here to 1.5 nanometer to 2. So we're changing just by half nanometer in size. And then we discovered that by actually just pointing a blue light into this gold quantum dots that are nano in size, just by changing the size, nothing else, no chemical or physical properties else were changed. We just pointed the blue light. This one had a different fluorescent colors that are pointing from them by just changing the size. And then this is have actually just when we realized we went to the smaller size, everything is being changing colors. All gold, all large gold that we see with our gross eyes is yellow. But when we went into a nano size, we started to see this. And this have been actually used in imaging technique. We are actually even using it on TVs. You could see that there's recently some new TVs company are producing anything with a QLED. They are using this nanotechnology technique. This is a new immersion. So in general, in simple, the smaller we went, we found that there is a whole different world of properties, reactivity, melting point did change, conductivity, color, gravity, all of that did have change. So what are the types of nanotechnology? So we have naturally occurring viruses is actually nanomaterial, but those are naturally occurring mineral composites and antibodies. All of those are nanomaterial just because they are fell into the nano size. There is a man-made incidental. We didn't mean to, like welding fume, sanding blast, and then the man-made engineering, the quantum dot, nanoparticles, and nanotube, which I will be talking about today. So, and a final part in how do we build nanotechnology? How do we create the nanoparticles? So initially there was a one way, the top-down approach. This is what used by all the electronic guys and the engineering. They start from a bulk big thing and we start chipping and chipping, etching them down till we go to the nanoparticle. This is how all people using processors and computer, this is how they go. Then now we recently discovered the new way. We started to go a bottom-up approach, meaning we start from an atom level and we start chemically and physically clustering them together till we reach the nanoparticle. This have exponentially expand our manipulation, and now we can actually create the materials the way that we wanted, going from the bottom-up approach. So that's our first part, where we just, now we have an intro to nano. It's a small size, different mechanics, different physics, different chemistry, and we now have an eyes and hands on how to manipulate, and we learn new techniques how to build them. So how did we apply those new techniques that we learned? So 2018, the Nobel Prize was shared by Dr. James Allison from Texas and Dr. Tasuku Honjo from Tokyo. Both have actually won the Nobel Prize for their work in immunotherapy. They actually created a new pillar in the cancer therapy by adding the immunocheckpoint therapy after we only had the chemo, radiation, surgery before. So Dr. James, he actually was the one who had discovered the CTLA-4 blockage, which is basically, in his research, he found that the dendritic cells or the antigen-presenting cells was actually not activating the T cells because of this binding of the CTLA-4. And only he created this blockage, the inhibitor. And so after he did this inhibitor, actually, he kind of removed the brake, and now the T cells are activated, killing cancer cells. So he just stopped the brake. This is just the basic science of checkpoint inhibitors. On the other hand, in Tokyo, Dr. Honsu have actually created the PD-1 inhibitor. So those PD-1 and the PD-1 ligand inhibitor, the pembrolizumab, but this time was immediately in the cancer cells, not in the antigen-presenting cell. So this PD-1 inhibitor from the PD-1 from the cancer cells was actually inhibiting the cytotoxic T cells, not to kill the cancer cells. And by creating this inhibitor, it kind of removed the brake, and now our cytotoxic T cells are actually attacking the cancer cells. And they won the Nobel Prize, a great, definitely improvement in the world of medicine. However, this was only effective on 15% of the patient. And there was a few challenges that we had to overcome with this immunotherapy. We didn't have enough accuracy, targeting, and specificity, neither the effectiveness as we could see in 15%. And here, where nanodrug do come into part, this is where we come into play. We're trying to improve all of those numbers, and we have actually able to accomplish. But in order to do that, we needed to learn some basic, at the microscopic level, we needed to learn the microenvironment. So we tried, first of all, we learned that the tumor microenvironment, a lot of papers have been published in the tumor microenvironment. We learned, first of all, the pH is actually more acidic around the tumor microenvironment compared to the rest of the microenvironment of the body. Another thing that we have learned, more classification to the time, we started to realize a great effect that where we can use the nanotechnology, which is called the enhanced permeability effect. So what's the enhanced permeability effect is, we found out that the endothelium cells in the normal tissue are normally attached, and the fenestration are very small. However, at the tumor tissue, we found the endothelium bonding are getting expanded larger and larger, which actually we started to use just by the size. No targeting have been used here. Just by the size of nanoparticles, they can easily infiltrate into cancer cells and not go into the normal vessels, just because of this EPR effect, the enhanced permeability effect. Because the size, they are more permeable for nanoparticles, and they go there. So this is the two actions, and now we will start with the application. So we have several nanoparticles that we create. All of those are man-made. Several forms, dendrimer, metal nanoparticle, liposome. A lot of them are being used now in actually not in clinical trials, but they got the approved carbon nanotubes, a single layer of carbons and graphene. But today I will be only focusing about actually two of them. Liposomes, which is being currently used in the vaccine of COVID-19, and we'll be talking about metal nanoparticle. So they have been recently just gaining a lot of momentum, especially after the two companies, BioNTech, Pfizer, and Moderna have used the liposome for their mRNA vaccine. And the other one is actually even CanSino, Oxford, and AstraZeneca are also using a virus-like particle, which is also a nanotechnology. People are saying, oh, I want to go with, I prefer rather the Oxford, but both of them are actually using a nanotechnology here. So this is just a quick about those three that I'm going to be highlighting today. Liposomes are less than 160 nanometer, have been used for several years in chemotherapy, and now it's being used in a vaccine carrier. Metal nanoparticles are going less than 80 nanometer, virus-like are also less than 100. So since 1995, we started to have the first approval into FDA approval of doxorubicin, where we started to use pegylated liposomes. Then by paclitaxel, attaching it to albumin particle in 2005, we got the approval for that. Afterward, the new treatment for ALL was also approved. And we actually have increased the half-life. We have decreased the toxicity from 25% to 13%. They had severe allergic reactions. At that time, when we added the albumin to the paclitaxel, we have actually even improved the effectiveness, and we prolonged the 5 years half-life for patients was 20% increased by just manipulating with the nanoparticles. Finally, 2020, right now, we're using the mRNA vaccine, which I will be talking about. But we'll go here. a little bit in step. So what's this liposome that have been used since many years and now have been used recently in this vaccine? So this is the conventional vaccine, since we learn into the medical school, is the bilayer of phospholipids, a hydrophobic and a hydrophilic. And then in the middle here, this is where they load their drugs, and they can load any material. For this example, they loaded the mRNA, but you can load even targeting material, chemotherapy, whatever is desirable can be created to be loaded here. Then in the top here, they have placed the polyethylene glycol, or what they call pegylated. This is to avoid the immune system, as when we started to place the polyethylene glycol, immune system do not attack our liposomes. Then here on the other side, we can attack any actually targeting protein that's desired. For example, sometimes we place the trastuzumab here to specifically target HER2 receptors. And also, we can add any imaging agent that can be seen in MRI or near-infrared imaging if needed, just to see where our liposomes are moving. So the size of liposome is 25 nanometer to 2.5, and they can be prepared in several ways, mechanical, solvent, or dispersion. We have been knowing liposome since 1965, but it actually came to practice in 1995 when we got our first approval for Doxil chemotherapy, and we are now advancing. Several companies have improved and manipulated the design of liposome. But what was interesting is the recent jump. Liposome, we have been researching for several years as an agent or a carrier for chemotherapy. Now it suddenly jumped to a vaccine. This is where it happened. So why liposome are being used as a delivery agent? First of all, they are biological compatible, low cytotoxic, and we can easily manipulate them. So why mRNA need the liposome? So mRNA in nature, we only have it inside of our cells. If mRNA is seeing extracellular, it will be immediately lysed by the extracellular ribonuclease. As soon as any cell dies and the RNA leaves the cell, it will be immediately analyzed by the ribonuclease. And that's why we have created this mRNA vaccines in the research lab since several years, but we were not able to deliver that mRNA to the cells. And that's where the nanotechnology come in place. We're just protecting and layering this mRNA in order to arrive. Several papers have been writing about this mRNA and nanomedicine, how they can improve the vaccine. So I will be talking in details about the BioNTech Pfizer vaccine. This is their general schematic. So they basically, in simple, they took the virus and then went into, by the bioinformatics, they took the messenger RNA that is responsible for the spike protein. They have actually took that specific part, incorporated into the liposome nanoparticle, injected, voila. Now, in their research, the BioPfizer, they actually have the 3 different trials. They were investigating a uridine mRNA, nucleoside-modified mRNA, which is the one that we ended up taking, and the self-amplifying mRNA. One of them was only a 1 boost. Both of them are prime and a boost, versus this one was actually a 1 injections. And they were seeing that the one that we finally have decided to go with was inducing more of a CD4 cells, meaning more antibodies than cytotoxic, versus this one was more of an immediate reaction cytotoxic. But why was the difference? Why did we need to go into 2 injections rather than the 1 injection? We'll go into that. But what part did they code it as? This S or spike protein of the coronavirus usually get into the endothelium cells by the ACE receptor. So this is the interaction that they wanted to stop. So they took the mRNA that is responsible for the spike protein, which was here encoded. And they decided, should we go with just the receptor binding domain, just this part, or should we go with the whole spike protein messenger mRNA? And they actually ended up going with the whole spike protein. So this is a comparison when they tried a 1 injection versus 2 injections. So if you get one injection, they decided that you could have a protection immunity when you reach a number of around 100,000 antibody IgG in your system. So after one injection, few people actually do reach the protection immunity. But however, when they give the boost one, they actually are reaching around 95% of immunity for patient. But when we gave only the amplifying mRNA with one injection only, actually a lot of people do have the immunity, but we are not reaching as high as the 2 injections. So even one injection do cover around 85% of people, but we wanted to improve the efficacy of those vaccines by giving the second boost. And this is basically it. So we have this vial, and the reason that we need to keep it so cold, just we are afraid that the liposome do lies at the normal temperature. And if the RNA again is released after the liposome, it's not toxic, but the ribonuclease will immediately make it ineffective. This is how we started to see in an electron microscope. So again, this is just an explaining or a recap of this spike protein that we started to see into the virus. We took a messenger RNA for this spike protein. And as soon as we had this messenger RNA, we were not able to administer it to the cell. And this is where we started to incorporate our messenger RNA into an encapsulated liposome. You could see easily the liposome do go inside the cell, and immediately our riposome simply started to produce the spike protein part. The spike protein will be easily exocytoses into the system, and now by the antigen-presenting cells seeing those spike protein will be creating antibodies. Antibodies will be released into system, attacking the spike protein. And this is just the spike protein. We don't have the virus. But now we are prepared. By the time we see the coronavirus enter the system, we actually do attack it, neutralize it before it even reach the ACE receptor into the endothelium cell. And this was simply the thought or behind it. So this is how we have used the liposome nanoparticle incorporation with the mRNA vaccine. I will give another example of nanoparticle that's being used, which is the metallic nanoparticle. Metallic nanoparticle can be anything, can be silver or gold, zinc or silica, and basically are created in something in a form called a quantum dot. Quantum dot is few atoms in size, have a core, shell, and a protective. The whole quantum could be from very few nanometers in size. Doxorubicin have been enhanced. This is an interesting or fascinating what they do with the doxorubicin. They actually have made it into three things, what they call is multifunctional nanocarrier. They use either the iron oxide or the gold nanoparticle. They made it for three things. One, doxorubicin was cancer targeting, improved to be made as cancer targeting, controlled delivery, and in imaging modalities. I will explain what they did in exactly, but they have seen after incorporating this multifunctional nanocarrier, they have seen that time after injection, we definitely are having a higher retention, and this is the basic of it. So this is a metal nanoparticle. Basically, this outer part is the trastuzumab they created on the outer side. Trastuzumab will be specifically to the HER2 antibodies that we see with the breast cancer. So this was placed for specific targeting. We will not send doxorubicin to normal cells, it only to specific to breast cancer who showed the HER2 antibodies. They also placed the chemotherapy, which we needed, the doxorubicin, and it was attached by some nanogold and linkers. So the center of this is a nanogold particle. This is the metal part, nanogold, which is only a few nanometers in size. And then they placed the polyethylene glycol just to avoid immune system. So this is how they went into it. Several companies have done this. They use either an iron oxide. So the nanoparticles are going inside, which we have placed with targeting material. it will go specifically to the breast cancers. And why is that? First of all, the EPR effect, the enhanced permeability that make this small size will go more retention into the tumor site. When they reach there, this targeting material that we created, the trastuzumab, will be specifically to the HER2 receptors and not to the normal breast tissue. So that's number one. We have the targeting, which will induce the endocytosis. Now, the whole compound of nanoparticle will go inside. By degradation of liposome, this nanoparticle will release the doxycycline, more shrinking the breast cancer. Now, keep in mind, the core of this nanoparticle is gold. So we actually can use this for all imaging modalities. We can use an MRI or a near infrared to make sure that most of our nanoparticle chemotherapy was really, and to confirm that it was actually deposited into the breast tissue and not scattered throughout the whole system. So again, this is a multifunctional. We had specific targeting with the trastuzumab by placing targeting. We had an imaging modality by just having the gold on top of the doxorubicin that we actually have created in the middle. So, and we actually have already played with this therapeutic load that was the center of the nanoparticle. You could use a doxorubicin, carboplatin, and paclitaxel. All of those are FDA approved and now into the market. We can even use etanercept. Nucleic acid can also be either the silencing RNA or DNA. In terms of targeting, whatever antibodies that we have specific, we can create the targeting material. Trastuzumab in this case was the one. We can even use different receptor ligand. And now we come into the part. So this is my third part. So the first one was where we started to see how is the nanotechnology, basic physics gave us these properties, gave us the privilege to do more manipulation. So we tried to improve several chemotherapy, several immunotherapy in that. And now we went into but with that being said, we are now facing the several toxicity, but we are not really able on how Dept these toxicities are going. We have very limited data as this is a new technique emerging. This advantage that we are really glad and optimistic about can become also a potential lethal factor. But again, nanoparticle at this point, it's only used in cancer and immunotherapy, and now we are advancing into a pandemic. So initially the discussion was, oh, we are already dealing with cancers and that are not, when we were weighing the benefit versus risk, nanoparticles in cancers patients were definitely winning because at least we didn't know the long-term toxicity, but we know those patients are really in a late stage of their disease. But the things the properties that made us happy with the nanoparticle, including the small size, can be dangerous as those nanoparticles can easily translocate within the body from an organ to organ. It definitely can cross easily the blood-brain barrier just because of their size properties. The silver nanoparticle that we use as a topical agent have been found that it induce and increase the ROS level and oxidative DNA damage, causing several DNA damage, but it needs a certain level to reach that. And that's why they're saying the topical we're using less amount, making us avoiding this oxidative DNA damage. So it's very limited. This is a new thing. It will not be the first time that we have created something to learn out several years later that we are missing few things. But we definitely know the cytotoxic damages have been tremendous. In conclusion, there's a lot of things that could be said good, and there's bad, there's ugly, but we are definitely happy that we are achieving an unprecedented clinical success. Despite there's a large fraction of patients are still not responding to immunotherapy, but the numbers, and we are really happy that we can advance quickly at this time. This new drug deliveries and emerging are coming, but I definitely want to point out and highlight that we need, as physicians, because this is a new technology. By the time I was in medical school, we only had less than 2 pages about immunotherapy teaching. So as a physician, we definitely need to be aware of what we are giving to our patient in terms of that. What's the side effect? What is the toxicity of that? Keep in mind that we need to know all forms of nanotechnology. They could come in a vaccine, in a topical agent, in a chemotherapy. So it's not only specific to rheumatologists or oncologists. We're talking about all physicians should be aware about this. Another thing I will highlight that in previously that in order for us to create any new material into the market, the average was 12 years, what they call bench to shelf. From the bench of the lab where we are designing the material into the shelf of the pharmacy, that process was in average take 12 years. But now, because nanomaterial is actually, we're using the same material that other people have already had the FDA and the safety profile. So when you're using a zinc nanomaterial, another companies have used zinc material. So we can go with that patent of those companies and we say, oh, now we're using the same material, but we're just making it smaller. So we might have that advancement and we can push for a quicker process. But we definitely are facing several obstacles. Mainly the toxicity we'll be aware about, and that's the summary of it. Thank you very much for listening, and I thank my program directors, associate program directors, and my mentors of oncology. I believe I also have Dr. Santra from University of Central Florida. Nanotechnology is joining us in case if we need any answers. Thank you all for attending. If you have any questions.
Well, thank you, Anas. That was very comprehensive and very topical, actually, given the vaccines that are available. Quick question about polyethylene glycol and some issues around allergy. I know those are definitely parts of the liposomes, these lipid alcohols. Any comments on that, and whether there's growing data that there may be at least some folks that have specific allergies here?
Nothing that I'm aware of. I looked into this. I've seen that there is those population who have it already, but I really don't have any specific data on that. Dr. Santra, are you aware about anything in this topic?
No, I'm not aware of any allergic action on PEG, but it may depend on the impurities because it's a man-made material. So depending on which company is selling and then the purity level, if there has been some contamination during the process, that could trigger allergic action. But as such, I'm not very familiar with the allergic aspect of PEG.
Thank you. Do you have anything also to add in general about this talk, as we have you with us, Dr. Santra, and nanotechnology in general, and what would you advise physicians in this topic?
Well, I would say, sorry about that. I would say that, first of all, thank you. It's an excellent presentation, Anas. I'm glad that you put together such a wonderful presentation for everyone to at least be aware of what is going on. As you know, that nanotechnology is still emerging, in particular in the medicine area. And applications, you mentioned the vaccine delivery. So certainly there are plenty of rooms to improve. One challenges that we always face, being a chemist or material scientist, I see that every size of nanoparticles is a different material, because the moment you change the size, it's not the same material anymore, because the surface property changes, surface area changes. So the narrow distribution of particle size is always preferred in some aspect, in some applications. And in some other applications, maybe a broad distribution of particles may be suitable. So having said that, the community should be aware of specific applications, and then a team of researchers should be involved. That includes doctors, scientists like us, and then the industry people, from the beginning, because that way we minimize our time for the development with a specific target application in mind. So those are the areas, including toxicity, including what could go wrong, because the major issue that I see when I design something, when we design something new from a material science side, we have limited knowledge what the clinical practitioners or MD folks will be thinking about, because we don't have knowledge to understand the inside our body what could go wrong.
Mm-hmm.
So that's why having a teamwork at the very beginning with a very specific problem in mind, the technology to think about this. Thank you.
You're cutting out, doctor, but thank you. I think I got the point that, yeah, definitely the whole collaboration between different interdisciplinary team is something very important. But thank you. Any questions? I see someone raising their hand.
I think Dr. Lawrence or Salam.
I can go. So my question is, excellent presentation, Anas. I can't describe how well your presentation was, but my questions are, so one is, when we're using this liposomes, we're saying that that's the reason that they need to be in an extreme temperature, and then they if they're not in that extreme temperature, they can lyse and hence release the mRNA. So the question is, A, how long is the how long will they need to be out of that deep temperature for them to start to lyse, or how, when does it become unsafe? And the second question is, does it apply for immunotherapies as well, or is this specifically for the mRNA coronavirus vaccine that we're using currently?
Thanks, Dr. Tuwaldi. Appreciate your feedback. Now, for the exact timing for liposomes in this mRNA vaccine, I really don't have the data. Although this is very important questions, I was not really able to find, at least in the data, that the BioNTech company. I looked into the BioNTech company. They were not able to see that, at least. But I know in different. And the answer to the second question is, have they been able to use this liposome in immunotherapy? Yes, they have done that. Several now immunotherapy are trying to be incorporated in that. And I actually know the data behind the time of liposome for other cancer therapy that have been using. Now, just keep in mind, there's several techniques how they can create the liposomes, and how is the liposome being made, as there's different type, and they can make them to be depends on what they want to do and which cells they are trying to get them to. So for those, the other that I have read about, they can actually go up for a few hours in that. So it depends what they are, how did they create it. I didn't go into the details how was the way that they form this liposome specifically for the Pfizer company, but I know for others that they can go, at least the one that I can aware about, they go for several hours.
Thank you.
You're welcome.
Dr. Raja, I see you raised your hand. You have a question? I think you might be muted or... Okay, any other questions from the audience in the meantime? I think in answer to your question, Salam, the two vaccines, the Moderna doesn't require quite the deep temperatures that the Pfizer does, and I assume it's probably the composition of the liposomes, right, on us that
yes,
are more stable. They don't completely lyse as soon as you bring them up to temperature. They do have a few hours that they remain stable. But I guess probably the composition of the liposomes or the Moderna vaccine are more stable at more reasonable temperatures than -70, I suppose.
that's true.
Abhishek, you have a question?
I do. Hey, Anas, that was a really, really good presentation. You kind of explained a lot of the intricacies and details of how drugs are really thought about and developed and envisioned. question. I don't know if you know the answer to this. J&J, Johnson & Johnson, just announced that they had some data on their single-dose vaccine, but they can keep it at 2 to 8 degrees Celsius. Do you know how that's different, or why that's able to be kept at a higher temperature?
So I have seen, I'm really not aware about Johnson & Johnson. Thank you, Dr. Kala. I wasn't aware about Johnson & Johnson. I have went into several companies that have been creating the vaccines, and I have seen that maybe they are not using the liposomes, because I have seen different companies that are using either the virus-like particles, and if you use that technique, or then you're able, actually, you're okay with temperature. They are very resilient, those virus-like particles, when you use them against temperature. The liposome have this bi-layer of phospholipid that are thermosensitive. I am not aware if they use the liposome that they are made thermoresistant. There is a company thing called Geo something that created actually a cancer immunotherapy, cancer therapy several years ago, that is resistant with the liposome that are resistant to temperature. But I'm sorry, I wasn't aware about this Johnson & Johnson. But definitely we'll look into it. Thanks, Dr. Kala. Dr. Palmers, I think you're on mute.
Is there any truth to the rumors being spread that the second dose of the Pfizer vaccine might be held off so that the rest of the population go ahead and get the prime? Or is it just a hearsay? We are hearing that in the press also.
Well, I have actually seen an article about this while I was preparing for this. Again, this is out of my league, but I have seen that several people are just saying because of the delay that we're having with manufacturer, that they are arguing whether or not we're going to the data that giving that only one part of the vaccine is giving some sort of an immunity. They said, what if we can give just one dose and increase the people who's being vaccinated? But again, this is not being made on a pharmaceutical level. This is not a recommendation that's coming from BioNTech or Pfizer or Moderna. This is a recommendation that's coming from an administrative level, and people who's just in the health department are arguing the benefit and weight of this.
In answer to Abhishek's question, I just looked it up. The J&J vaccine is an adenovirus vector vaccine. It's not a liposome, so probably that's why it's a little more stable. It's more of a more conventional technology.
Mm-hmm. Thanks, Dr. Palmer.
Any other questions from our audience today? All right, then. Well, Anas, thank you for enlightening us. It was a really terrific talk. Very interesting. I may try to get a hold of some of those cartoons. We can show them in the vaccine clinic when people ask, how does this thing work? Yeah. It was very helpful to...
Yeah, definitely. What actually induced that when people, and I initially started this as a joke. A lot of people on Facebook or social media are saying, there is when they hear the nanoparticle part, they are worrying about the chip and a thing, and we actually need to do that. I'm hearing this concerns actually from people who's in the paramedics, and so we definitely need to educate people, because people, when the social were asked about what's nanotechnology, 55% of the American population have said, please, you scientists, do not play with God work. When they were asked about specifically nanotechnology in medicine, it actually reached the support for that for 88%. And that's why actually I'm happy to share those videos, if we can educate the people and even the physicians and my colleagues just in that regard. So, thank you all.
Thanks, everyone. We'll see you next week.
Thank you all. Thank you very much all for coming. Have a good day.